Hello, everyone. Welcome to my video tutorials for spatial data analysis. Today, I'm going to show you how to analyze the CODEX data using Giotto package. Giotto is a R package that has been developed to analyze spatial dataset. If you use this package to analyze your data, please acknowledge the authors who developed this package and also cite their original papers. The dataset that I'm going to use for today's demonstration is the mouse spleen dataset. If you want to use this dataset for practice, you can download the data using this link. First, we need to install the Giotto package. I installed it already, so we just need to load the Giotto library. So the Giotto package requires the Python environment for data analysis. To use the Giotto package, you can set your own Python path. Another possibility is to set the Python path as null. Then you can install the Giotto environment on your computer to use the Giotto mini conda environment. You only need to do this once. Next time, when you start to use the Giotto package, R will automatically detect the Giotto mini conda environment. So if you are the first time to use the Giotto package, you need to run this code. So I don't need to run this code anymore. We can go straight to the data analysis. So the first part of today's demonstration is to load the data and create a Giotto object. Then we process the data. For spatial data analysis, we need the expression matrix data and also the cell coordinators data information. For this dataset, we also have the metadata, so we can load the metadata into R as well. So first, we can use the read expression matrix function to load the expression data for the mouse spleen dataset. So let's load the matrix data first. You can see here in the data window, we have a large DGC matrix dataset. We can convert the expression data to a matrix data and have a look at the data information. Let's convert it. Then we can click the matrix data. So you can see in the row names, we have 30 proteins. And the column names are the cell numbers. You can see here, we have in total 83,786 cells. Then for the expression matrix, it is the expression level for each protein in each cell. So next we can load in the cell coordinator information. The cell coordinator information is a text file. It will be in the cell locations folder if you download the data from the link I showed you in the beginning. We can load the cell coordinators as CODEX locations. And we can click the data and have a look at the cell coordinators. You can see we have the first column for the cell IDs. The second column is the X position and the third column is the Y position for each cell. Because we also have the metadata information for this dataset, we can also load the metadata. So let's click the metadata and have a look at the metadata information as well. You can see the first column is the image phenotype cluster ID, the second column is the cell ID, the third column is the sample styles. So in this dataset, we have 62 tiles, which means the whole image was taken by 62 times. Then we have the X tile index and the Y tile index information for each tile. After that, we have the niche cluster ID, we have the X and the Y position. In the last column, we have the cell type information for each cell. So as I said, we have 62 individual tiles in the metadata. Because we want to analyze the whole data on one image, so we need to combine all the tiles into one image. In order to do that, we first need to set a global XY coordinators to stitch the tile coordinators. For this dataset, we can set the X tile span as 1344 and the Y tile span as 1008. You can try different settings. If you set the numbers bigger than the settings we set here, you will get the gaps between each tiles. So we set the global coordinators. Next, we can stitch individual tile coordinators under the global coordinators to form one whole image. So I showed you already the tiles position are in the metadata, and here is the global coordinators. So we can use the stitch tile coordinators function to stitch 62 tiles into one image. We can run. So next we can switch the CODEX location for each cell to the global coordinators. Before we do that, we can have a look at the CODEX location dataset again. You can see at the moment it's the X and Y position for each cell. If we switch their position to the global coordinators, let's run. And we can have a look at the CODEX locations file again. You can see now it is the X and the Y global locations for each cell. So we got all the data ready. Now we can use the expression matrix data and also the global cell coordinators to create a Giotto object. Because we have the metadata for this dataset as well, we can also include the metadata. So we can use the create Giotto object function to create a Giotto object. You can see we created a Giotto object and named the CODEX test. You can see down here, because I set the Python path as null, so when we create the Giotto object, you can see there's no external Python password provided. Then I use the mini conda Giotto Python environment to create the Giotto object. As I showed you in the metadata, we have the cell type information. But you know, for the immunohistochemistry, sometimes there are unspecific antibody binding. Those information was included in the metadata. We can use the unique function to have a look at the cell types in the metadata. We can run. You can see here, in fact, we only have 25 cell types. But when we use the microscope to take the images, we also get the fluorescence signal as unspecific binding. So those places was labeled as dirt, noid, or capsule in the metadata. You can see from the cell type names, we have the noid on 26, then dirt on 17, We also have the capsule on number 4. So because they are not cells, we can subset the data and filter out the cell IDs labeled as dirt, noid, or capsule. So first we can get the cell IDs that were not labeled as dirt, noid, or capsule. Let's get the cell IDs. Then we can subset the data to get rid of the cell IDs that labeled as dirt, noid, and capsule. You can see if we subset the data, we only keep 80,019 cells for our analysis. So now we can subset the object. If we have a look at the Giotto object now, you can see here we only have 80,019 cells now. So next we can filter out the low quantity cells. So there are different thresholds we can set for the filtration. First we can set the expression threshold as 1. Then we keep genes that were detected in minimum 10 cells. And also we only keep the cells that have at least 2 gene expression. So I explained the threshold that we are going to use for the data filtration. So we can filter the object now. You can see here we only removed 2 cells from the dataset, and we kept all the genes because we have 30 genes in this dataset. If we have a look at the object now, you can see we only have 80,017 cells, but we still have 30 genes. So after the filtration, we can normalize the data. So the Giotto package provides a function called normalize Giotto to normalize the data. We can normalize the data now. So after the data normalization, we can add the gene and cell statistics into the object. So before we add the gene and cell statistics data into the object, we can have a look at the CODEX test object again. You can see we have the raw expression data slot to save the gene numbers, cell numbers, and the expression levels. We normalized the data already. We have the spatial locus data slot to save the cell coordinators. We also loaded the cell metadata. I showed you already the cell metadata. You can see we have all the cell columns names here. At the moment, we only have 10 columns. Then we also have the gene metadata. You can see in the gene metadata, we have the cell ID and the gene ID, but the rest data information was 0. So now we can add the gene and the cell statistics data into the object. We can run the add statistics function. Let's run. So we should add the gene and the cell statistics data in the object already. You can click the CODEX test object name again to look at the cell metadata and the gene metadata as I showed you just now. But Giotto also has a built-in function to access the gene metadata, this one called fDataDT. We can use the head function to look at the gene metadata. Let's run. You can see now, before we only have the cell ID and the gene ID in the gene metadata, but now we add the cell numbers that express for each gene and also the percentage of the cell numbers that express for each gene. We have the total expression for each gene, name, mean expression, and the detected mean expression. And also we can use the pDataDT to look at the cell metadata. Let's run it again. You can see before the last column is the image in phenotype cell type column. Now we add the total number of genes information in each cell, and also we add the percentage of genes in each cell, and also the total expression for those genes in each cell. So we normalize the data and also added the gene and the cell statistics data into the object. So now we can use the adjust Giotto matrix function to adjust the expression matrix. In this step, for the expression matrix adjustment, we can minimize the technical variables batch effect that could affect our data analysis. So let's adjust the expression matrix now. So we created a Giotto object for second part of analysis. Before we proceed to the second part of analysis, we can have a look at the dataset. First, we can use the spatial plot function to look at the cells. Let's run. We can zoom in. You can see here the global cell coordinators. We set it in the beginning for X coordinators and Y coordinators. We stick all the tiles. We have 62 tiles here into one image. You can still see the gaps between each tile. And also we can set the cell color as sample X tile and Y tile, and use the spatial plot function to look at the tiles. We can run. Let's zoom in again. You can see now each tile was labeled as a different color. We have 62 tiles here for the whole image. So this image could help you to understand why we set the global X coordinators and Y coordinators in the beginning, and also stitch all the tiles into one image. Because during the data acquisition, the microscope take the image one piece by one piece. Then for our data analysis, we want to put all the small pieces together as the whole image to analyze the data. So you can see here the color labels each small pieces. I showed you we also have the cell type information in the metadata. We can also set the cell color as cell types and plot the cell types on the image. Let's run. Let's zoom in again. You can see now we have 25 cell types here, and you can clearly see distinct cell clusters on the whole image. So now we finish the first part of our analysis. So next I can show the second part of our analysis. In the second part, part 2, we are going to perform dimensional reduction, cell clustering, and cell types visualization. So the whole process of this part of analysis will be very similar to our single-cell RNA sequencing data analysis. So normally in single-cell RNA sequencing data analysis, we need to find the variable features first. You can also do this for Giotto object. Giotto package has a function called calculate highly variable genes. Because in this dataset we only have 30 genes, so we want to use all the genes as variable features. So we don't need to run this step. You can find the gene names in the dataset I showed you in the matrix data. We have 30 proteins as row names. We can get the proteins name from the row names of the matrix data we run. You can see here are the names for 30 proteins, and also you can get the protein name from the gene ID in the CODEX test object. If you run, you can see all the proteins name are the same as the row names of matrix data. So if we don't run the function calculate highly variable genes, then when we run PCA, we will use all the genes as variable features. Let's run PCA now. So you can see down here, highly variable genes was not found in the gene metadata information. So all the genes were used for PCA analysis. So after PCA analysis, if you want, you can plot a PCA. Let's run. You can see here is the PCA plot. The x-axis is the PCA1 and the y-axis is the PCA2. We can also run the significant PCA function to identify the best PCA numbers for downstream analysis. Let's run this function. You can see we generated two figures. Let's zoom in again. You can see the first one is the percentage of variance explained per PC. If you look at the curve, it's very similar like the elbow plot. When we select the best PCs for single-cell RNA sequencing data analysis in red, you can see the first 5 or 6 PCs could catch most of information. And the second figure is the cumulated percentage of variance explained. You can see if we just use PC1, we only capture like information below 25%. If we increase the PC, if we goes to like 15, 16, it's almost 80%. For PC 27 and 28, it's almost 100%. You can see from the data, if we use like PC 15, 16, will be good enough for our analysis. But if you want, you can also use until PC 30. For today's demonstration, let's just use PC1 to PC 16 to run the UMAP analysis. We can run. So after second dimensional reduction UMAP, we can create a nearest network. This is a similar function as find neighbors when we use the thread package to analyze single-cell RNA sequencing data. So by default, if we run the create nearest network function, it uses the shared nearest network function. If you want to use the k-nearest network function, you can set the type as KNN. So we can run this function now to create a shared nearest network for our demonstration data. After the nearest network analysis, we can find clusters. So Giotto package has 4 choices you can use. You can use the Levin method, the k-means method, the hierarchical clustering method, or the Nathan method for cell clustering. If you use Levin, you can use the function do Levin clusters. If you want to use the k-means method, you can use the function do k-means, and do h class is the function for hierarchical clustering. For today's demonstration, we can use the do Nathan cluster function To find the cell clusters, let's run this function and name the cell clusters as laden. So after the laden cluster analysis, we can get the metadata again. We should have a new column in the metadata because we named it as laden. We can click the metadata now. You can see we have 14 columns now. You can see in the last column, we have the laden cell cluster. Here is the cluster number for each cell. So now the data is ready for visualization. First, we can use the plot UMap function to plot the cell clusters. Let's run. You can see here is the UMap for the cell clusters. We can zoom in. You can see in total we have 20 cell clusters here. And you can see each cell cluster has been shown by a different color. So we identify the 20 cell clusters with the resolution as 0.5. So now we can use the spatial plot function to look at the localization of different cell clusters on the image. So we can run the spatial plot function now. And we can zoom in again. You can see the localization of each cell cluster on the whole image. If you like, you can also visualize the cell cluster and the cell clusters on the image. Let's run. You can see we created the image to show the cell clusters on the left-hand side and also the location of each cell clusters on the image on the right-hand side. So we finished the second part of the analysis. Now we can move on to the third part. In the third part, we can perform differential expression analysis to identify mark genes for each cell cluster. Then you can use the mark genes identified by the differential expression analysis to annotate the cell types. So in the Giotto package, there are two methods you can use to perform differential expression analysis. The first one is the Spleen method. This one performs pairwise the t-test. And the second method called Gini coefficient method. Because we identified the cell clusters, use the laden method, and the cell cluster information is in the laden column in the metadata. So we can set the cluster column as laden. Then we can use the find markers function to identify marker genes for each cell cluster. Here we are going to perform each cell clusters against the rest cell clusters. So for today's demonstration, I'm going to use the Spleen method. If you want to practice the other method, you can set the method as Gini. Then we are going to use the normalized expression values, and we identify minimum 3 genes for each cell cluster. So let's run this function to perform the differential expression analysis. We can run. You can see R start to identify mark genes for each cell cluster. It is very quick. We finished it already. And it generated a data frame we named as markers Spleen. Let's click this data frame and have a look. You can see in total we have 191 observations. So you know in our dataset we only have 30 genes in total. If we click the genes column, you can see the same gene was identified as a mark gene for different cell clusters. because later we want to create a heat map for each cell cluster to show the best marker genes. Then we need to set the gene name as row names. You know, in the row names, we are not allowed to have repeat names. So we can use the unique function to keep just the one gene name for the heat map. Let's run the unique function. Because we named it as mark genes screen now. You can see here we have 30 genes. So that's all the gene names we have for this dataset. So now the gene names are unique to the row names. We can create a heat map to show the mark genes for each cell cluster. Let's run. Let's zoom in again. You can see here is the heat map for the mark genes. You can see the best mark gene for the cluster A will be the CD44 and the CD169. The best mark gene for the cluster 19 is CD19. You can see for cluster 1, the best mark genes are 480, CD106. So this cluster should be macrophages. Another example is the cluster 15. The best mark gene is the NKP36. So this cluster should be NK cells. This is the heat map for the marker genes for each cell cluster. If you prefer use the wiring plot, we can get the top marker genes for each cell type. Let's run. Then we can create a wiring plot to show the best mark genes. Because we are going to create a stacked wiring plot, we only show the first 8 marker genes. Let's run. We can zoom in again to see the wiring plot for the 8 marker genes. You can see if 480 is high in the cluster 1, so cluster 1 should be macrophages. And you can see the ERTR7 is highly expressed in cluster 7. So cluster 7 is the cell cluster for stroma cells. So you can use the marker genes we identified by differential analysis to annotate the cell types. Because I showed you in this dataset, we have the metadata for the cell types already, so we don't need to annotate the cell types. If you want to annotate the cell types for your own dataset, you can use the annotate Giotto function to name the cell types for your dataset. Okay, that's it for today's demonstration. I showed you how to use the Giotto package to analyze the CODEX dataset. Previously, I also created videos to show you how to analyze the CODEX dataset using the thread package. So I hope my video tutorials can help your spatial data analysis. If you like my video tutorials, please subscribe my channel and share my videos with your friends. Thank you.